this journal club, I think, will be a little bit weird because a lot of it will be about history and why we are where we're at. And I'm happy to take lots of questions about that. I wish Gabe was here. I think this is a topic that would be very relevant to him, of a history.
I wanted to see you guys argue about it.
Yeah, I mean, I think we'd be mostly in agreement because, I mean, this is history. This is what happened. So there's that. So to start off, both of these papers are pretty old. They are, one's from 1986, and one is from 2003. So they're at least about 17 years old. And they were extremely influential to the field of synthetic biology and to the field of building cells. And largely wherever synthetic biology has kind of touched, like the discipline of synthetic or bioengineering, they've also been very relevant. And they're kind of papers that you might not know about, that were buried quite deeply in the literature, and that I've only really found from talking to the people who are involved in making that history. So let's take an overview. Let's rewind from now to back then. So in 2016, the first synthetic cell was created by the JCVI, called JCVI Syn3, which could be a whole different paper journal just on that technology. But the general idea is they synthesized a minimal synthetic cell. It was a huge accomplishment. Through the 2010s is kind of when industrial biotechnology started taking off, with the founding of Ginkgo Bioworks in 2009, which is now a $1 billion company. Back then, they had founded Ginkgo Bioworks to get better at engineering life for people, generated on the hypothesis that there was more people who needed to bioengineer life than the market currently served. And if they only could serve those people, we could get great leaps and bounds in biotechnology. In 2005, the iGEM competition was kind of started by folks at MIT. Those same folks went on to be the founders of Ginkgo Bioworks. But iGEM itself was started back there in a response to some stuff from the synthetic biology study. In 2004, AdGene was founded, which is the largest plasmid repository currently available. And, it and iGEM are basically the only plasmid repositories currently available for biotechnology. In 2003, kind of concurrently with the synthetic biology study, the BioBricks assembly method was published, which was a standardized method for assembling DNA with standardized parts, which laid the foundation for more of the advanced techniques for standardized DNA assembly. And so far is still the largest and most well-known DNA assembly method, even though it's pretty much obsoleted. That leads us to our 2003 synthetic biology study, which is actually not the paper we're going to start with. And this synthetic biology study is important for one really fundamental reason. It made a plan. It made a coordination solution about what we should do to get better at engineering biology. And that was super important. In 1997, Drew Endy, professor that I work under, and that wrote the synthetic biology study in 2003, got his PhD in simulating T7 on a computer. And Drew Endy and Tom Knight, who was another professor at MIT, went on to kind of write the synthetic biology study and do all the synthetic biology things at MIT that laid the groundwork for modern-day synthetic biology. And so Drew's PhD was on simulating a T7 genome on a computer. Both Endy and Tom Knight, who were pretty much the founders of synthetic biology, were both inspired from basically one man, Morowitz, and what I think is his best paper, The Completeness of Molecular Biology, which will be the first paper we'll be starting out with. I don't really know how this normally goes, but has everyone read the paper? Like, looked it over? Should I talk a little bit about it?
Yep,
What is-
I've read it. Or both of them.
I have also read it, and most people are supposed to read it when joining the club. So if they haven't, then I would just start with the assumption that most people have read it. But if you want to give a brief overview, that would probably help as well.
Yeah, so the important part about this paper is that it made an observation, and it put forth with like this kind of basic researcher attitude the idea that there is a completeness of the logic of life is finite, which is an extremely fundamental kind of idea that he proposed that hadn't really been proposed before, and which inspired a lot of the modern synthetic biologists. And I'd like to just, highlight a few things that he did here that were really important, and then we'll have like a question session for anyone who's like wondering about this. The first is that he did like a back of the hand calculation on synthetic cells and how small they could be, which I would encourage everyone to do if they're doing any biology. It's like the Glowing Plant Project didn't do this, and how that ended up. He was remarkably close with how small the smallest cells were, so about 700 kilobase pairs and 600 proteins. The smallest cell we have so far is about 500 kilobases and 473 proteins. So he was remarkably close with his back-of-hand calculations. His ideas on which genome to use, mycoides, were actually spot on. That is the one that the 2016 Synthetic Life was based off of. And he, in his paper, exactly says, like, we should use this one. And his 600 steps computer model is what inspired Drew Endy later on his PhD. And probably, if I had to like go back and say the most important part of this paper, it's that the quote that's important is the existence. Let us return to the mycoplasms as the simplest living cells. Their existence, with all the properties of life, say that the logic of life is finite, relatively simple, and subject to full exploration. That is probably the most important part about this paper. And there are other things to read into, but this was kind of the founding text that set synthetic biology into motion. Any questions on that one?
I think we have a lot of people here, and I assume because of the silence that some people may not understand biology. I know from the last week's journal club, some people were in a completely different field. So Keone, could you maybe do like the most bedrock of introductions to what synthetic biology means?
Sure. Yeah. Thank you for that.
Cool. Thanks.
Yeah. So synthetic biology is kind of a field that was based off of bioengineering, and specifically genetic engineering in the bioengineering field. And the basic premise of synthetic biology is that if we get really good at engineering life and engineering biology, we can both learn fundamental things about the world, so basic science, and also we'll get better at engineering biology. Kind of the idea of, in order to understand something, we have to be able to create it. In synthetic biology, the endpoints were about those two things. And how it got there was rigorously applying engineering concepts to biology, which previously hadn't really been done. And so the hope was by specifically applying them to biology that we do a lot better at engineering it.
Cool.
Oh, and also, Keone, I know that you said that you worked for Drew-ND, but do you want to give a little bit more of a background about what you're currently doing before we dive a little bit deeper into everything?
Yeah. So, sorry for not giving an introduction. Hi, I'm Keone Gandel. I currently work at the Free Genes Project at Stanford University under Drew-ND, which is a project to make DNA free and open source for the world. You can imagine free and open source software, except now we have free and open source DNA. Drew is who I work under, is a professor at Stanford University, who was previously a professor at MIT. And he was one of kind of the foundational figures in the founding of the field of synthetic biology, and is just a very eccentric and interesting person to be around. And, my history has been 3 years at doing biology for the Free Genes Project, as well as mitochondrial engineering and directed evolution work at UCI. And then I've also done a couple of years of biohacking. Any other more basic background questions that anyone has?
I'm thinking about your professor. Was he helping with the mini PCR project?
I don't think Drew was involved in the mini PCR project.
Okay, thank you.
Yeah, that was some different professors.
And you said he was the first to ever model the T7 genome, which is one of the most basic versions of a virus, correct?
Yeah, so it's a well-known and well-studied virus. It's not the simplest. It's about 40 kilobases. And kind of the goal with that was to put to test Morowitz's hypothesis that life is finite and we can simulate it on a computer. It was a test of that hypothesis. Turns out the answer was you pretty much can't. It was too complicated, which was one of the things that led up to the synthetic biology paper.
Okay, cool. Do you want to start getting into the second paper?
Yes, this one is probably the more important one for a lot of people.
Just to double check, the second paper is the Morowitz one?
The second paper is the 2003 synthetic biology
Gotcha.
study.
With our boy, Drew-ND.
Your boy, Drew-ND.
Yeah. So, Keone, as a general question, what was the initial response to this paper when it came out in 2003? You've mentioned that this paper had a big impact, but what does that mean?
Yeah, so there are two different kind of answers to that question. Probably the biggest impact was it was the first paper to really make a plan for engineering biology, which was super important because it represented a bunch of smart professors and PhD students at MIT making a plan for a field, and this plan was flawed in a couple of ways, and we still feel kind of the shock waves of the parts that they were wrong about. But this plan, in a lot of ways, was correct, or it was the right way to do things. And the cool thing about making a plan and kind of spreading it out is that policymakers and people and funders can look at this piece of this paper or this presentation or this study and say that's the plan we're going to follow. So it gives policymakers credence to fund certain activities, which they did. The other thing that it did that was interesting is it created kind of a coordination solution, a way of thinking about the world that a lot of people in the local area could adopt at MIT. And so, for example, people involved within this study were the ones who created iGEM and created AdGene and created the BioBricks standard. And so a lot of the things that they talk about within this study were things that immediately afterwards actually went into effect because people saw an opportunity because a plan was made and they could coordinate labor and capital around doing the plan. And as far as I can see, there has not been a similar, plan that has been produced or a similar form of leadership in synthetic biology since this paper.
Yeah, I was just going to say too that it's interesting because when Keone first introduced this to me, I was like, oh, there's no way. There's like this secret club of people that basically founded the entire field and control most of the policy nowadays. But you look into it and there's basically just a small group of guys that met up for this study and a few other studies and kind of laid the groundwork for the entire field. And now a lot of them, even while working their normal jobs like Drew ND, Church and all of those head figures are also involved in a lot of like boards that, aren't even really publicly recognized sometimes. But like I know we've talked a little bit about like the advising boards for synthetic biology to like some of the US agencies before and stuff like that, and how all those guys usually sit in and get to say their feelings on stuff like that, even though they might not be heard some of the times. But
yeah, that's a great point. A lot of the people who originally acted on this like idea or were part of it are now the people who have all of the soft political power. Drew ND, George Church, Tom Knight, the founders of Ginkgo Bioworks, Addgene, iGEM. Those are the major synthetic biology institutions. So it really was like a small group of people who just wrote a plan and then started implementing the plan and were given all of the capital and political resources in order to try and make that plan come true.
It's like the Federal Reserve, but for biology.
Yep.
Yep.
And those people are now on all the boards of well-known synthetic biology companies that are getting like billions of dollars funded into them.
Yeah.
Suffice it to say, plans are important. And so this plan in particular was created at MIT, where basically what happened was, from Drew's words, is that he decided to talk about the future of bioengineering with a bunch of different graduate students or professors or interesting people that were at MIT. And on Tuesdays, they would have free burritos and free tacos in a certain room where they would talk about this thing. And it turned out that small amount of food was sufficient to drive, most of the people who are interested in this type of thing at MIT to come talk to them, which is how they founded a lot of the ideas that are discussed within this paper. One very interesting part that I should preface it with is that a lot of the people that they attracted were actually from the electrical engineering fields. A lot of people from electrical engineering at that time at MIT got involved with the synthetic biology study. And this might have just been random. It might have been selected somehow. I'm not sure. But that did happen, which had a massive influence on synthetic biology in the future. So let's start off with any questions on that? Please shout out any questions because I like talking about them.
So if there's so many electrical engineers that were, giving talks, listening to talks, attending these small gatherings at the beginning, early 2000s, I assume that sort of framework for thinking, life is just circuits and flow diagrams ingrained itself into thinking about synthetic biology. But the framework, the way that we think about stuff also impacts how, we process problems. Do you feel that people or researchers today are too locked into frameworks thinking about biology in terms of circuits and flow diagrams? Or do you think there's still enough flexibility that people aren't getting stuck on these analogies?
I think that they, right now, are more stuck than they think they are. Because of like the idea of thinking about it as circuits and stuff has kind of just been propagated. And the ideas about kind of circuits have proven to mostly be wrong in biology. And so there's a change happening. You could probably identify it happening around the early 2010s when people stopped thinking about DNA as much as circuits and more about like DNA and its whole complexity and all its many interactions. But still, you will find that you'll still find people and still find places that teach biology and bioengineering very similarly to how the original synthetic biologists taught it. It's still very influential, but it is changing. So,
Yeah, I was also going to comment that it's quite interesting seeing how like those two mentalities kind of play out in just the biohacking space in itself, because you have a lot of people that are pursuing projects and they're like, oh yeah, no, it's super easy. I can just slap a plasmid in that boy, and then all of a sudden, boom, project done. And then the project turns into like a 10-year thing, and nobody can figure out why it doesn't work. Like the insulin project and all of those things. So it's also kind of interesting because there are some type of projects, like for example with Justin and Gabe's lactose intolerance, where it's like there are studies that have been going on for a very, very long time that can significantly prove that if you use these specific components in just this specific order, then the thing will work. So it's more of like an engineering approach than a kind of like electrical computational approach that seems to work the best for getting things done from what I've seen in the biohacker space.
Yeah, and like the idea that biology is just a bunch of circuits that can be put together like a computer program was pretty much largely made in this study and turns out to not really be that true. But people still take that analogy to heart.
Cough, cough, Josiah, cough, cough. Yeah.
Yeah.
From what I understand, one of my friends has the ability to poke at databases and understand, of course, there is a big need of understanding how to make a protocol behind a plasmid creation, and how do you actually from the melting point and so on. That how would you figure out how to do it? But there is